Welcome to the fun that is aerobic respiration. So I just want to put in a disclaimer right now. I apologise for if I end up messing up and saying oxidised instead of reduced or reduced instead of oxidised. I'll try and put like a pop-up on the screen if I say the wrong thing. It's just when I'm speaking it doesn't process through my head, like what I think in my head doesn't come out in the words, so I may say like the wrong word. So I apologise, I will try and fix it. But to start off with, let's figure out how we make ATP through respiration. So your first stage is glycolysis. I've sort of simplified the stages down in this, so I'm skipping out some of the intermediate molecules. To start off with, you have your glucose molecule in glycolysis. So your glucose molecule actually dephosphorylates two ATP molecules, forming 2-ADP. This means that the glucose molecule gains two phosphates on it, and this forms something after it sort of reorganises itself called fructose 1,6-diphosphate. So basically a glucose molecule with one phosphate on one end and one phosphate on the other end. But this is very unstable, so it splits into two molecules. One is called GP and one is called DHAP, but these are interchangeable, so most of the time you just call it GP. This original splitting of the fructose molecule was done by enzymes, as you probably guess. So next up you've got two NAD plus molecules being reduced into two NADH molecules. This oxidises the GP molecule, and then there's a lot of intermediate stages as well, but I'm skipping over them. You've also got 4 ADPs being phosphorylated into 4 ATPs because you've sort of got phosphates in the mix there. Altogether, this then forms two molecules of pyruvate or pyruvic acid, which can then enter the Krebs cycle. So yes, I'm calling this the Krebs cycle. It does have a couple other names, such as the tricarboxylic acid cycle or the citric acid cycle, and it happens twice per molecule of glucose because you get two pyruvic acids. So to start off with, you have your pyruvic acid. This combines with a molecule called coenzyme A and loses a carbon dioxide molecule, so decarboxylation, and it also causes NAD to be reduced into NADH. Altogether, after this little process bit of it, it forms acetyl coenzyme A. So this acetyl coenzyme A then enters the cycle and combines with a 4-carbon molecule and loses the coenzyme to form a molecule called citrate. This then undergoes oxidation and decarboxylation, which means electrons reduce the NAD plus into NADH and a molecule of carbon dioxide is released. I've not included it on the diagram here, but the product from this step, I can't pronounce its name, it's like alpha-ketoglucaric acid, I think, actually recombines with the coenzyme A, but it's lost in the next stage, so I didn't really include it because I didn't really need to know it. But it goes through the same process as above, so it is oxidised and NAD plus is reduced back into NAD plus is reduced into NADH, and it's decarboxylated, so it loses a carbon dioxide molecule. This forms succinyl-CoA, but I'm not including the CoA in this bit because it is lost in the exact next step, where a phosphate group is substituted for the coenzyme and a high energy bond is formed from a GDP molecule to a GTP molecule. You then have FAD being reduced to FADH via the combination of two hydrogen atoms. And you also have another reduction of NAD plus into NADH, resulting in that 4-carbon molecule that we started off with combining our acetyl-CoA with. So our net products from your Krebs cycle, I nearly said carboxylic acid cycle then, that's not right, is 6 NADH, 2 FADHs and 2 ATP per molecule of glucose. Final step is the electron transport chain. This occurs in the mitochondrial membrane of eukaryotes, but occurs just in the normal membrane of prokaryotes because they were once mitochondria. This works off a basis of redox reactions, but first off you have NADH being oxidized into NAD+ because it's donating its electrons to a membrane-bound protein called FMN. This also pumps hydrogen ions from the matrix into the intermembrane space, creating a gradient of H+ ions between the two membrane areas. The next protein, which is called protein Q, causes FADH to be oxidized into FAD, so again it loses some hydrogen atoms there. And this receives electrons from the FMN protein and passes them on to the next protein in the chain, which is a cytochrome complex. These electrons continue to move along each cytochrome complex in the membrane until they reach a special one, but each time the electrons passed along it loses a little bit of energy. So let's talk about this special one. So as I mentioned, there's now a ion gradient there's a H+ ion gradient. There's more H+ ions now in the intermembrane space than what's in the matrix. This means that they have to diffuse down an electrochemical gradient, and they diffuse through the cytochrome A3 complex. Here, these hydrogen atoms are combined with an oxygen molecule, 1.5 of oxygen molecules to form 3 H2O molecules. At the same time, these hydrogen ions are moving down the electrochemical gradient through an enzyme called Atp Synthase, and as you can probably guess by the name, it's responsible for ATP synthesis. So it uses the water molecules that we've just made, hydrogen ions and NADP to form ATP. And through this it can form around about 32 ATP molecules, which is a whole lot more than the measly 1 and 2 we were getting in all the other stages. So at the end, through the use of one glucose molecule, we get around about 36 ATP molecules from aerobic respiration.